By Professor Ralf Heine, entitled Evolving Our Understanding of the Impact of 2FL and LNT on the Gut Microbiome and Metabolome in Infants with Cow's Milk Allergy.
Thank you again for the kind introduction. My task is to now provide some mechanistic background to the observations that we found regarding the infection reduction and what's actually happening with the microbiome in infants receiving HMO supplemented formula. I think breast milk is the gold standard of infant nutrition, and it provides a good guide for any future development in the formula space. So, and we know that the metabolic and microbiome effects of breast milk are largely driven by human milk oligosaccharides, and there's hundreds of them, different kinds of human milk oligosaccharides in breast milk. These are very, very important in establishing the early microbiome towards an infant-type microbiome, and this is a point I want to drive home, and I will explain this in greater detail. Until recently, infant formulas did not contain any human milk oligosaccharides, and it's only since they can be made via bacterial biofermentation that we have now the benefit of breast milk identical components that can go into formula. And this is also now followed by a rapidly expanding knowledge about what these actually do in vivo and in vitro. I want to just spend a little bit of time to talk about infant-type bifidobacteria, because this is a distinction that's quite important. The infant-type bifidobacteria you can see at the bottom here are basically 4 species that are very good at assimilating and utilizing HMOs. So the main one, the first one, Bifidobacterium longum subspecies infantis or Bifido infantis, is the all-round master at utilizing HMOs. But the other 3 that you can see there, B. breve, B. bifidum, and B. longum longum, are also able to utilize bifidos and are typical in the early colonization of the infant microbiome. You can see that these bifidobacteria have specialized transporters that allow to assimilate HMOs, and then have a specialized set of enzymes to break down these HMOs into smaller ingredient sugars, if you like, which are then fermented in the bifidobacteria with the outcome of mainly lactate and acetate as the main metabolites. And it's now very important to understand that acetate and lactate can also be then cross-fed to other bacteria which co-establish in the gut early on. And I just want to maybe draw attention to the conversion of lactate to propionate by Veillonella, which is an early colonizer. And also, very important, the conversion of acetate to butyrate. And you've already heard in the talk by Professor Omani that butyrate has very strong tolerance-inducing and immune-modulating properties directly on cells, but also on barrier function. You can see in the right text box all the different effects that butyrate has on the colonic epithelium, but also on tolerance development. And this is the set of enzymes that we don't possess. So these are glycosyl hydrolases that are bacterial. And you can see that we would struggle to do any of these digestive steps in our gut, but with the help of the bifidobacteria, we can suddenly utilize HMOs via bifidobacteria. And you can see that breakdown products also can be utilized by other bifidobacteria. And once one sort of infant-type bifidobacterium has established, you can see that based on the microbiome, on the HMO breakdown products, other bifidobacteria follow, and they also freeze out other unwanted bacteria by competing around nutrients, and many bacteria can't utilize these breakdown products that you're seeing up above. There's also a new discovery on aromatic lactic acids, and these have previously been unknown to be of importance, but these are important immune modulators for tolerance development, and they modulate T-cell responses and monocyte responses. And they are coming from aromatic amino acids, and then an enzyme that was previously unknown until a couple of years ago, aromatic lactate hydrogenase can actually make these different types of aromatic lactic acids which have immune modulatory function. Now, coming back to our studies, you've heard very nice summaries of these studies. So the first one was the cinnamon study of an extensively hydrolyzed formula with 2 HMOs, and you can see here that it was predominantly a growth study, but that we also saw a trend in reduced respiratory infections. And this is mirroring the findings from a healthy infant study also, which showed a significant reduction in lower respiratory tract infections. The stool collection schedule is shown here. So they had a baseline sample collected, the infants before the intervention, and then follow up 1 month later, another 2 months later, so at 3 months follow up, and then all infants had a final specimen taken at 12 months of age. And you can see the numbers here of the infants initially randomized, 97 in each group. We had follow-up later to a smaller group of infants where we also looked at metabolome. Looking at what bacteria were actually enhanced or enriched in the microbiome between the two groups, we could see that bifidobacteria and infant-type bifidobacteria here in yellow were preferentially enhanced. You can see, for example, other bifidobacteria, which we also know are beneficial, but for example, here at the lower end, Bifidobacterium adolescentis is not enhanced because it's not utilizing HMOs. So the HMO effect really selects out the right bacteria, bifidobacteria, for the infantile period. We then also looked at how the microbiome evolves over time, and this is done by a so-called cluster analysis where you look at what stages the microbiome goes through, and I won't have time to go through this in detail, but you can see here on the right panel the early colonizers, and you can see that we have sort of 10 or 12 main colonizers, and the gray area above are all the other bacteria, and as the infant ages, diversification happens. We then followed these community types over time, and you can see fecal community cluster 1, 2, 3, 4, and 5, and infants usually go through these towards a more stable fecal microbiome pattern. And we could see, when you can compare the control versus test for each column, that there were significant differences. And basically, what we did is we split the cohort into 2 infants, because we had enrollment ages from 0.5 months to 6 months, and that to look at microbiome is a huge gradient. So to get a clearer picture, we looked at those enrolled early, under 3 months, where complementary diet is not confusing maybe the findings, and then the later enrolled group, where complementary diet also may modify more importantly. And you can see that we have significant differences, and that the infants who did not receive HMOs ended up with a more adult-like pattern around 12 months. And if you look at the yellow bars here in the early enrolled or in the late enrolled infants, you can see that about 50% of infants in the non-HMO supplemented group had reached an adult-like pattern, whereas only 25% in the HMO supplemented group. And you could argue, why is this important? But if you stay for longer in a bifido-dominated microbiome pattern, you may also prolong the window for early immune modulation. So this may actually be additional time they have to develop normal immune responses. We also looked at metabolites, and acetate, as I showed earlier, is one of the main metabolites. But the signature was maybe not as impressive as we were hoping, and the reason for that is multiple, but we can also see that, for example, the control infants already start at a very high baseline, which indicates that some of them would have had breast milk exposure previously. So it's not a black and white picture, but what we could see is that in the control infants, the early acetate signature goes down, whereas in the HMO group, acetate goes up. And that is almost significant in this setting. And we have to admit that fermentation of dietary fiber at the later visits probably obscures the HMO effect. And that's a technological limitation that we have because infants obviously commence a diet, and dietary fiber is also fermented. So we see a competition of fermentation products. And this is even more complicated, and I won't have time to go through it entirely. But what we looked at is the 16 key metabolites that were altered between the HMO supplemented and non-HMO supplemented formula, and also 17 KEGG orthologs, which are the enzyme pathways that these bacteria have. And you can see all the different enzymes on the right side, and at the bottom you see the metabolites. And what we saw is two main pathways, apart from the acetate that are short-chain fatty acid pathway. We had an effect in the bile acids, and we could see that conjugated bile acids were decreased. And we also could see that in the amino acid pathways that there was decreased breakdown of branched-chain amino acids and aromatic amino acids. So we saw a downregulation of oxidative protein breakdown. What these mean in a clinical setting, I think, needs to be explored, and I can just offer this as a description of what's happening, and I think it needs further interpretation what this means at the clinical level. The second study, the platypus study in Australia, and we're lucky to have some of the investigators here in the audience, was primarily a growth study, but we also looked at microbiome outcomes. It was a single-arm study of about 30 infants with moderate to severe cow's-milk allergy, and it was a mix of a few infants with IgE-mediated features and the majority of non-IgE-mediated features. Again, we did a detailed shotgun metagenomics analysis, so all the speciation of the bacteria, plus also a quick look at short-chain fatty acid profiles, but not as detailed as for the previous study. And the visit schedule, again, quite similar, a baseline sample, a 1-month follow-up, 4 months, and then a 12-month sample. And I show you here now firstly to illustrate that infants, even at baseline, already had different patterns. And if you break down the baseline sample by age, you can see that there's already a transition to different patterns. But what you can see is, if you look at the top level, the actinobacteria in light green, and at the bottom panel, the bacteria, Bifidobacteriaceae, in light green as well, you can see that the space that they occupy at baseline is not very big. So there's a depletion at baseline of Bifidobacteria. And if you then look at visit 1, you can see that the light green space in both graphs has expanded, which means that there is a bifidogenic effect, so an enhancement of infant-type Bifidobacteria in response to HMO feeding. When we looked more in detail around the HMO-utilizing Bifidobacteria, there are two groups. There's the infant-type Bifidobacteria, so the 4 that I introduced on the earlier slide. There are also 2 others, Bifidobacteria, Pseudocatenulatum and Catenulatum, that can both utilize HMO and dietary fiber. So they're more transitionary Bifidobacteria, which appear towards the end of the first year. And you can see that we had early amplification from baseline to visit 1 of the probably early infant-type bacteria, and then later on a further strengthening of the signal of bifidobacterial enhancement, which also includes the strains in under 5 and 6. So we do see an enhancement, and I also want to draw your attention that some infants at V0 did not have any bifidobacteria that could utilize HMO, and could also not amplify bifidobacteria as we went along. And this is an important fact to also bear in mind, that some infants have a very deranged microbiome and may not be able to utilize HMO anymore. Finally, we did a taxon set enrichment analysis, and that compares just which bacteria are the most enhanced or the most suppressed, and I won't have time to go through this in detail, but we saw that bifidobacteria, Lachnoclostridium and Bacteroides were generally enhanced, and you can see at what levels that was significant, whereas some abundances, for example, E. coli and Klebsiella, which are proteobacteria, were reduced. So we see a picture evolving of increase in early stage bifidobacteria or HMO assimilating bifidobacteria, and a reduction in proteobacteria, which is really the opposite of what describes the dysbiotic stage. So we see a partial correction of microbial dysbiosis. And just finally, very briefly, we could not distinguish fiber effect from HMO effect because it was a single-arm study, but we can see that the infants had a significant increase in acetate from visit 0 to visit 1, which could be attributed to an HMO effect. And we also see that butyrate and propionate rise later on, hopefully, or most likely, due to a mixed fiber and HMO effect and cross-feeding, for example, of acetate into butyrate by butyrate producers, which we also saw emerging, and I won't have time to go through that in detail. So in summary and in conclusion, we saw in infants with cow's milk protein allergy that breast milk identical HMO promoted an enrichment in infant type bifidobacteria, and the stress is really on infant type, so these are very specific HMO utilizing bifidobacteria, and a reduced abundance of proteobacteria, suggesting a partial reversal of the dysbiosis that we see commonly in cow's milk allergic infants. We also could show that HMO supplementation slowed the premature shift towards an adult type microbiome, and we saw some metabolic signatures changing, mainly regarding fecal acetate, but also bile acids and amino acid breakdown. But clearly we also need further well-designed studies to look at these immune modulating effects and metabolomic effects in greater detail. So there's a lot of work ahead of us. Thank you very much.